All right, so let's go ahead and move forward with part two. We're going to wrap up the Rosaria here. We have one group left to go, and this is the Circus Owens. So again, remember Rosaria, pseudopods for movement is the key feature for Rosaria. Within Rosaria, we have the radiolarians, and then we have the foraminifera and the Circus Owens. So only three subcategories of Rosaria. So a lot smaller amount of content to worry about here. So, all right, but Circus Owens. So what's going on with this group? What's unique? What's different? Again, remember, they have to be a radiolarian or Rosaria to be here. Once you have Rosaria going, to be a Circozoa, they have a few other features we want to talk about. So one of the features is they can also use flagella for movement. So they have the pseudopods, but they also can have flagella that help them move. So that's a little bit of a different feature than what we saw with the radiolarians and the forames or foraminiferas. So flagella here. Location-wise. These guys are going to be found in terrestrial environments, but I'm working on my spelling here. They need to be in moist terrestrial environments. So this is not part of the plankton load in an aquatic ecosystem. These guys are terrestrial organisms, but moist terrestrial. So not likely to find them in a desert, but if we dig down into the soil in our backyards, very possible we may come across these guys. You're going to need a microscope to see them, though. So what is their role? Well, these guys are photosynthesizers, but it's because they have endosymbiosis. No, I really am off on spelling today. Sorry about this. So they're endosymbiosis with green algae. So again, technically, is the Circozoan running photosynthesis? No, it's the green algae that lives inside of it. So do we truly give the Circozoan credit for photosynthesizing? Well, officially, you would give the credit to the green algae, but that's the tough part with classification and organizing life. You can make that argument about a plant and say, well, it's the chloroplast that technically runs photosynthesis. So, but anyway, we're going to call these guys part of the producers or part of the producer load in terrestrial environments. So that's all we're really worried about with Circus Owens. So single-celled, flagella for movement along with pseudopods, terrestrial environments that are moist, and endosymbiotic with green algae. So it makes them a producer within a terrestrial ecosystem. So, okay. All right. That takes care of Rosaria. So let's do the review slide. So you got Rosaria. Can you tell me what is the key feature to go in Rosaria? So what feature do I need to be in this supergroup versus one of the others? Now, once I'm in that supergroup, what makes a radiolarian different? What are the features here? What are the features for foraminiferans that make them different? What are the features for Circozoa that make them different? Can you fill this out? If you can, awesome. You're going to hit a home run here. If you can't, practice. Print off the slide. Practice. That's why I'm building review slides in all of these at the end of every super group, is for you guys to get a feel for where you at with this. Do you remember all these features? I mean, there's a lot to remember. Do you have all that in your head? If so, great. If not, practice and practice and practice, because you're going to need to know this for the exam. So use the review slides in that fashion. Okay? All right. So now let's tackle the Amoebozoa. Give them what color? Let's go with purple. All right, Amoebozoa. Supergroup Amoebozoa. This is a fun category, very fun group. Within it, we have the Amoebozoa. So you kind of go, makes it a little easy to remember this one subcategory. Now the negative, there's gonna be 3 different groups or organisms that I talk about that go within the Amoebozoa. Okay, so what makes an Amoebozoa do its thing to make it unique and different? Well, let's red star this. Let me get that one. Red star. Big key feature for the Amoebozoans. They have oh, sorry, I didn't realize I would kick it over like that. All right, so Amoebozoans. The big feature, they have pseudopodia, just like the Rosaria. But the pseudopods have actin and myosin filaments within them. So if anybody's taken anatomy and physiology, or you're going to, actin and myosin are these little protein structures. Little protein filaments that give them structural durability to it, a little more structural integrity compared to what we see in the Rosaria. Hence, you notice in the Rosaria, everybody had a shell around them. Amoebozoans, they don't have a shell or a test to help give them structural durability because the actin filaments are more durable. So these guys are kind of like jello with little fibers infused into their cell membrane to give them, like I said, some durability to it. So within the Amoebozoans, the 3 groups that we're going to talk about, the amoeba, the Plasmodial slime molds, and the cellular slime molds. Okay? So those are going to be the 3 subcategories of Amoebozoans that we're going to tackle. There's some really neat guys in here, and I'll share a little story with you guys about what happened to my house a few years ago. So, okay, so let's check out the Amoebozoa and say, okay, what's going on with this particular category of Amoebozoa? So amoebas. These organisms are single-celled, free-living organisms. They're found in all sorts of environments. Let me fix free-living first. Probably do that. They are found, or can be found, in moist terrestrial environments and freshwater ecosystems. We do not see amoebas in salt water. Because if you threw one of these guys in the ocean, there's a thing called osmosis. They cannot regulate salt concentrations, and literally all of the moisture in their body would get sucked out because of osmosis if they're placed in a hypertonic environment, right? So it's kind of like, hopefully nobody's done this, but if you guys ever put salt on a slug and it sucks the moisture out of the slug's body and kills it, same thing would happen to an amoeba if it was exposed to a salty environment. But moist terrestrial environments, they need to be moist and wet all the time. Unfortunately, some of these guys can be parasitic. Parasitic. So here's a bad part. Some varieties will move up to your brain and parasitize and basically eat away at neural tissues. That goes in the bad category. Most of these guys will just act as decomposers. Where they recycle nutrients. Others, let me move this guy. Others will act as omnivores, where they literally eat anything that they come in contact with. Okay, so here's a little video of what it looks like when amoebas are moving. So you can see them just kind of oozing in different directions. So the oozing is called cytoplasmic streaming. And what they're doing is literally oozing cellular structures throughout their body. They're pushing their pseudopodia this way. Oh, I don't like that. Then they may contract or they may extend. Notice how it's getting pushed up here. That is the basic movement of the amoeba. Now, if they can find something to eat, they push those pseudopodia and they wrap it around the organism. They literally wrap themselves around it to engulf it through a process known as phagocytosis. Okay, so here's a phagocytosis. Those little guys zipping around are paramecium. Hopefully it sounds familiar. And notice the stealthy attack of the amoeba. It has just wrapped itself around the paramecium. The unsuspecting paramecium. I need that trailer voice you hear in the movies. The amoeba wraps around, and now the paramecium is trapped inside. And, oh, I can't get out. And now notice the amoeba is just going to collapse that little bag or whatever you want to call it that wrapped around the paramecium. It just keeps closing it in on it. And what's going to happen is eventually it's going to release digestive enzymes. And oh my goodness, there we go. It's horrible. Cover an eye if you don't want to see it. That's what they do when they eat. So phagocytosis. Oh, I got to get all these out of here. So, okay, let me pause and get back to our lecture. Whew, okay, that exhausted me. So those are the amoeba. Good guys, bad guys, part of an ecosystem. But this is one of those things you want to be careful if you're in any kind of aquatic environment that's not the healthiest and you have parasitic amoeba in there, it can be bad. But overall, there are more good amoebas than bad amoebas. We do want these guys in our ecosystems. So, okay, let's take a look at the Plasmodial slime molds. These are a crazy little creature that traditionally was grouped, classified differently a few years ago, but now we've reorganized them because they have the key amoeba or amoebozoan feature, the pseudopodia made of actin and myosin within their body. So the Plasmodium, this orange, what looks like an orange net, that is actually the body of the organism. So we call it the Plasmodium. And it's this multinucleated, or nucleated will make more sense, multinucleated mass of cytoplasm, meaning there's a whole bunch of nucleus in there. It is one cell, but it contains a bunch of nuclei. So that's the weird thing. Instead of just a single nucleus in a single cell, you got one big, gigantic, massive cell, cytoplasm, that has a bunch of nucleus within it. So weird guys. Ecologically, they're going to eat things like bacteria. yeast, basically microorganisms in the soil. So these are terrestrial. We're not going to find these in aquatic systems. Moist as well. You get dry, doesn't do them any favors. So moist environment. When it does dry out, okay, so when it does dry out, or these things go through stress, and stress could be temperature drop, like winter's coming, drought. It's moving into July in Illinois, and we don't have rain, and they're stressed because it's getting dry and there's no food. They form a sporangia, and that sporangia are these little stalk structures with the little bulbs on it. Almost looks like blueberries there on little sticks. And those sporangia release spores. Those spores then move or get blown or released further away from the parent organism. And the idea is that they will begin a new generation further away from the parent, hopefully in a better environment. So, okay, let me pause here. Again, trying roughly 15-minute window break. And then I will show you an example of, I had one of these on my house years ago. And then we'll get into the last member of Amoebozoa. And I want you guys to think of Power Rangers, if you remember that from your childhood, as we go into our last member of the Amoebozoas.